Hello and welcome everyone. In this podcast we're going to talk about eukaryotic transcription. Up to this point we have discussed the basics of transcription, and then we talked about transcription as it relates to bacteria, and now we're going to focus on eukaryotic transcription. And like we did with the bacterial transcription, I want to begin by talking about promoters and RNA polymerases and how they differ between bacteria and eukaryotes. This time I would like to begin by talking about RNA polymerases. In bacteria, if you recall, I guess we can write this down, bacteria, there was really one RNA polymerase. And the thing that varied with the RNA polymerases to give it some sort of specificity was that sigma factor. In eukaryotes, we know there are at least 5 different RNA polymerases. I'm going to actually say 5 plus because I want to talk about something else with the mitochondria and chloroplast. The first one is RNA polymerase 1. Second one, RNA polymerase 2. And RNA polymerase 3. And these next two I'm going to lump together, RNA polymerase 4 and 5. RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 2 transcribes genes that code for mRNA. RNA polymerase 3 transcribes tRNAs. And then 4 and 5 are only found in plants. I should point out that 1, 2, and 3 are also found in plants. These are found in all eukaryotes, and plants being eukaryotes, they have 1, 2, and 3. However, plants have additional ones, 4 and 5. And there may be more out there that we just haven't discovered yet. These plant ones, I want to say just one quick thing about them, is they code for siRNA, these interfering RNA molecules. And their job is more of a defense. By degrading viral RNA. So an RNA virus attacks a plant cell. The plants have this mechanism of having this RNA polymerase 4 and 5 produce this RNA molecule that will bind to the viral RNA and then degrade it. It's a much more elaborate pathway than the way I just described it. But for now, just know that these siRNA will bind to the viral RNA and target it for destruction, and thereby protecting the plant. 1, 2, and 3 here are RNA, mRNA, tRNA. The way I always remember this is that RNA polymerase 1 codes for rRNA, and R has 1 hump, if you think of this lowercase r, and 2 codes for mRNA, and the M has 2 humps, so that helps me remember it. And RNA polymerase 3 begins with a T, and tRNA begins with a T. Just a goofy way I remember it. And the reason I say 5 plus is because eukaryotic cells contain mitochondria, and they contain chloroplasts. Each of these contain their own DNA. Not surprisingly, they have their own RNA polymerases. They don't use the ones we just spoke about. Interestingly, the chloroplast one is related structurally, functionally, and by sequence. It's related to a bacterial RNA polymerase. It's maintained that one ever since it was taken up into a eukaryotic cell. Now, interestingly, the RNA polymerase found in mitochondria is most closely related to RNA polymerase found in bacteriophage, both structurally and by sequence. There's just one more thing I want to mention about RNA polymerases, and that is the type of RNA polymerase found in archaea. Archaea used to be lumped in to a larger group, prokarya, with bacteria. But we have since learned that archaea are more closely related to eukaryotes than they are to bacteria. Even though archaea and bacteria are both microorganisms, evolutionarily speaking, they are quite different. And even things that occur within the cells of archaea are much more like eukarya. As evidence to this, the archaea RNA polymerase is much more similar to eukaryotic RNA polymerases, and less similar to bacterial RNA polymerases. Alright, that is all I want to say about RNA polymerases for now. I want to now move on to promoters that are found in eukaryotes. So let's go ahead and draw the promoter region of a eukaryotic cell. We're going to write the ORF here, the open reading frame, and we'll indicate this as the start site here, start of transcription. TXN for the abbreviation of transcription. So this is +1. We then have this core promoter. A very important consensus sequence of the core promoter is found in this region here, that is at -25. 25 bases from the start of transcription. This site we often call the Tata box. This is called a Tata box because the sequence here, the consensus sequence, is TA, TAA. There are going to be some variations to that, but in general it will be very similar to this sequence, and so it's called a Tata box. And what happens, and we'll see this on the next slide, so I don't want to spoil it for you quite yet, but a protein will bind to this region here to initiate transcription. But we'll talk about that in a little bit. This Tata box in the core promoter region is all you need for basal transcription. And when we say basal transcription, what we're talking about is a very low level activation of this gene at this open reading frame. So it's not tightly regulated using just this core promoter. Now eukaryotes will have an additional sequence here, which we're going to just call for now a regulatory sequence. Now what binds here are one of two classes of protein, and depending on which sequence it is will depend upon which proteins bind. But it could be an enhancer, or a silencer. These are great names because they tell you exactly what they do. Proteins that are enhancers, they will bind to the regulatory sequence, and when they're bound, they will allow the transcription from this gene to be much higher than basal. The silencers do the opposite. They silence the transcription. So when a silencer protein binds here, it will silence this. Now something that's misleading by the way I drew this is that these regulatory sequences are typically fairly far away from this gene. They're usually somewhere about the position -50 to -100 base pairs away. However, they can be even further away than that. And so it's interesting to try to figure out how a protein can bind here and somehow affect the RNA polymerase that's binding here. And to explain this, I'm going to erase this bottom part of this whiteboard. And I'm going to explain what happens, and then I'm going to try to draw it. You have to remember that these pieces of chromosomes are extremely flexible. They're very dynamic. They're not these very static and immovable structures. They're very flexible. And so what happens is when an enhancer or silencer binds here, the chromosome here will flip over here. So let's draw this, and I'm going to draw this right now as just a straight line. It'll be easier to draw. And we haven't talked too much about it, but when this RNA polymerase is bound here, and we're getting that basal transcription, very low level transcription, how can we make this go faster? And remember, these are about -50 to -100 base pairs away. Well, when an enhancer binds here, it causes a structural change of the chromosome, and the whole chromosome will flip around. And by the magic of time-lapse photography, quickly close your eyes and you'll see what happens. chromosome flips over like that, and now the enhancer, or it could have been the silencer, but in this case we're talking about enhancer, that was bound 100 base pairs away, now is right next to it. And it can interact with this protein here by either preventing it from going forward if it's a silencer, but if it's an enhancer, it can bring in necessary proteins to make this RNA polymerase move much faster. So these enhancers and silencers act far away, but have a great effect on the overall transcription of a gene. Okay, let me go ahead and erase this. Now I want to talk about the complexity of eukaryotic transcription. I want to be clear, I'm not saying eukaryotes are more complex. I'm certainly not going to say humans are more complex than, say, a fruit fly. But what I will say is that eukaryotes have a much more complex system of turning genes on and off. This is in part because we have more DNA. We can list some reasons here, more DNA. And in multicellular organisms, we have different cell types. We also have developmental reasons to concern. The heart has certain genes that are turned on as an embryo versus the genes that are turned on as an adult. Because of that, it's more complex. Which genes are you going to turn on and which ones are you going to turn off, depending upon which tissue you're in and which stage of development you're in. I always like to think of it as a symphony. I have zero musical skills, but I like to listen to music. So I know you can have a symphony here and a symphony here with the same kind of instruments in it. But this symphony may play Stravinsky's Firebird. If you've never heard it, it's a great one. Or it may play one of my favorites, the 1812 Overture by Tchaikovsky. Now each of these symphonies has the same number of instruments, the same types of instruments, but the conductor is directing a different kind of music from each one. The same tools are available in each one, but a different thing happens. Same thing with a cell. You could have a heart cell, and then you could have a pancreas cell. They each have the same number of genes in them, but different ones are turned on or off at different times of development and to get heart function versus pancreatic functions. Much like, I think, a symphony, where you have different music coming from two different symphonies that are, by all practical purposes, the same. So how do eukaryotic cells do this? Get different kinds of genes turned on and off. And they do this in several ways. We've talked about some of them, but certainly you think of the different kinds of RNA polymerases that bind. We talked about the enhancers and silencers. And what we haven't talked about yet are general transcription factors. And I should also say, put a fourth one here, other transcription factors. We haven't talked about these last two yet, but we will now, because they explain a lot about how which genes are turned on or off. Also, just to point out, the RNA polymerases bind to the core promoter, as do these transcription factors. They bind to the core promoter as well to regulate which gene is being turned on or off. So let's spend some time now talking about these transcription factors. And in doing so, let's talk about it as it relates to the first stage of transcription, initiation. Initiation requires RNA polymerase. Without RNA polymerase, you are never going to make the messenger RNA. However, RNA polymerase is never going to find the promoter without general transcription, TXN for short, factors. So these two are what are needed for initiation. Now there are other transcription factors that help fine-tune that regulation, but this is what we need for initiation. And there are specifically at least 5 general transcription factors that are needed. So let's draw our famous gene here with our core promoter here with that TATA box there, T-A-T box. And what binds first here to initiate, to get the whole system going, is transcription factor. It's a general transcription factor, transcription factor 2D. We say transcription factor 2 because it's RNA polymerase 2 that's going to be binding here. It binds to this top box. You also need to know that a particular protein that's found in this transcription factor 2D is called TBP, for TATA binding protein. It is what binds to this piece of DNA. Now there are other transcription factors that will now bind here as well. I want you to know two others. One is transcription factor 2H. It will also bind here. This one's important because it contains a helicase, and this helicase, just as we saw helicase is important in replication, this helicase will open up that DNA to allow for transcription. And then one more that binds in here that I want you to know, and that is transcription factor 2B. It comes in here as well. And the reason this one's important is because this is what ultimately the RNA polymerase binds to. Now when you're looking at this on the promoter, if you could see it with your naked eye, it would look like a big glob of protein stuck here. But they come on in a very regulated fashion, all 5 of these. You just need to know these few here though. The TATA binding protein with the TF2D comes on first. It recruits the other transcription factors, such as transcription factor 2H, which is important because of its helicase function. And then transcription factor 2B comes along, and it's important because it's what brings RNA polymerase into the show so that it can begin to transcribe and make RNA molecules. So that's really what initiation is all about. These proteins coming along and opening up the DNA so that RNA polymerase can come along and make a new RNA molecule. Now, as I said on the previous whiteboard, is you could also have these enhancers flipping over here too and interacting to initiate transcription as well. But without the enhancers, you will still get a basal level of transcription from this gene. Now, once elongation starts and the RNA polymerase starts zooming down here, these transcription factors now fall off. You'll still have a few other proteins attached to this RNA polymerase, but what you're looking at now is a complex of proteins that lacks most of these transcription factors, and it continues to make RNA as it moves down the gene. Now the last thing we want to talk about is termination. RNA polymerase 1, it terminates similar to having that rho-like protein. It's not a rho factor like we saw with bacteria, but it's rho-like. It's very similar. We're not going to talk about it, but I want you to know that we're not going to talk about it in great detail, but I wanted you to know briefly how it terminated. Then RNA polymerase 3 terminates after having a long string of adenine. So they pairs with the uracil, and that weak association allows it to fall off, similar to rho independent that we see in bacteria. Now this last one that we've been focusing on today is RNA polymerase 2. RNA polymerase 2 has to fall off, but just like we saw in bacteria, it doesn't know when to fall off. So let's draw our gene like so, and the gene codes for this area here. We haven't talked about it yet, but we will on the next podcast. That is a poly A site. So RNA polymerase has to make sure it gets past this before it falls off, and it does. So it keeps moving along. It passes this. The RNA that it's made now, like so, will have transcribed this poly A site, which is again very important for the life of the messenger RNA. But how does this fall off? Well, there are one of two models. The first model is called the allosteric model. What happens with the allosteric model is as the RNA polymerase is moving along in this direction, the RNA is cut off. When this happens, and what we call it an allosteric change, essentially the shape of this protein changes, in part because factors on this protein, which we call elongation factors, which as I recall, I don't think I mentioned when I talked about elongation, but I should have, or elongation factors, EF, that are bound here that allow elongation to occur. Once this cleaves off, it destabilizes the RNA polymerase, and these elongation factors now fall off. That causes the whole system to fall apart. The rest of this RNA that's moving along is released and is then degraded, and the RNA polymerase also falls off. And now the rest of the molecule here is free to go on and then be processed, which we'll talk about next on the next podcast. So we call that the allosteric model. The next model we want to talk about is called the torpedo model. What a great name. It's similar in some respects in that transcription occurs and it passes this polyase site, and then it is cleaved here. And this RNA molecule can now go on and be processed and start making proteins. But we still need to stop this RNA polymerase. And the way this happens is there's this protein called RAT. So RAT1 is the name of the protein. It binds to this end of the RNA molecule. RAT1 turns out to be a RNase protein, like we talked about when we talked about the Avery, McLeod, and McCarty experiment. It's an RNase. And so what it's going to do is move up towards this, much like our Rho factor did, but this is a little different because instead of chewing it up all the way here, it slowly just chops it to pieces. And once it gets to the RNA polymerase, it then breaks apart and releases the RNA polymerase and has degraded all of the RNA that's being made that's not part of the primary RNA transcript here. So both these models, the torpedo model and the allosteric model, are both viable models, and there's evidence that both of them exist, and that it's more likely that within any given eukaryotic organism that both of these actually occur. Okay, that ends the podcast. Let's go ahead and give a quick summary. In this podcast, we talked about eukaryotic polymerases, and we talked about 5 different ones we see in eukaryotes, plus some additional ones that we see in mitochondria and chloroplast, and then we briefly touched on the one we see in archaea. We also talked about eukaryotic promoters and how they are different, how they have the core promoter with the Tata box and then the regulatory sequence where the silencers and enhancers combine, and how that can raise transcription levels up significantly. We talked about how those transcription factors can bind to the core promoter to enhance this transcription. And then we talked about initiation, elongation, and remember when I talked about elongation, I failed to mention elongation factors. If you remember, I said that those initiation factors fall off, which allows the polymerase to move down the gene. What I forgot to tell you was that elongation factors also now bind to that eukaryotic RNA polymerase to move it down the gene. And then we close by talking about the 2 models for termination, the allosteric model and then the torpedo model. All right, that's all I have for this podcast. We just have one more podcast to talk about with transcription, and that's going to be something that happens specifically with eukaryotic RNA, and that is how it is processed. Okay, if you have any questions, let me know. If not, I'll see you in class. Bye.